So meiosis is a special type of cell division that creates eggs and sperm, and it's important to understand the steps that the cell has to go through to be able to divide into those special types of cells. Before meiosis can actually start, you have to go through a step of pre-meiosis replication, where the cell replicates its DNA and creates enough organelles and cell membrane to actually be able to divide into daughter cells. So once you've replicated all those organelles, like the endoplasmic reticulum, mitochondria, and the DNA, and you have enough membrane to divide, the cell will go through 2 rounds of division called meiosis 1 and meiosis 2. So meiosis 1 is broken into 4 phases. Meiosis 2 will be 2. And the first phase of meiosis 1 is prophase 1. So prophase 1 is usually the longest step in meiosis. It can take up to 90% of the time that's spent through the entire meiosis cycle can happen in prophase 1. So in prophase 1, if we're looking at a cell like this, with a nucleus and the nuclear material still all spread out in there, by the end of prophase 1, you've still got just one cell, but instead of having this nice intact nucleus, the nuclear envelope has started to break up, and inside the nucleus, the homologous chromosomes have all paired up. The sequences that they have in common actually line up with each other. The biochemistry of how this happens isn't really well understood, but it's a process called synapsis, and it's held together and helped along by some proteins called the synaptonemal complex. So these 4 chromatids stuck together by the synaptonemal complex are called a tetrad. While you've got all that stuff going on inside the nucleus and in the nuclear envelope, you also have a couple of little bodies forming out here called the centrosomes. And these centrosomes start to separate from each other during prophase 1. So after this prophase 1, the cell goes into metaphase 1. In metaphase 1, you don't have a nuclear envelope anymore because it's totally disintegrated. Those centrosomes have reached opposite sides of the cell. Like could have gone up here or over here. I'm just putting them side to side. And they've got these microtubules sort of spanning the entire cell like this, going out like that, sort of reaching into the cell and giving them some structure to work with. And all the homologous chromosomes in their tetrads have lined up along the center of the cell in what looks like a plate. So back in the 19th century, they started calling it the metaphase plate. So they line up a little bit like this. Here are a couple of more homologous chromosomes that I didn't have in that last step, but there they are. And the microtubules are attaching themselves to each of the homologous chromosomes at a place called the centromere, which is part of the chromosome. And once you've done that, you move into anaphase 1. In anaphase 1, you've still got the centrosomes hanging out at the poles, but the microtubules that they had attached to the chromosomes have started to shorten. And they're pulling one set of chromosomes to either end, like that. Now, which chromosome goes to which end turns out to be totally random. And that randomness is what gives rise to independent assortment. Okay, from anaphase 1, we go into telophase 1, which is going to be the end of meiosis 1. In telophase 1, the nuclear envelope starts to reform, the centrosomes have disappeared, and the cell is starting to divide at the beginning. And a lot of textbooks will show the chromosomes totally spreading out into unorganized chromatin again. They do a little bit, but really they just sort of, they only partially decondense. So I'll just draw them in here kind of fuzzy-like. And by the end of telophase 1, these 2 cells will have completely divided. They don't usually spend a lot of time waiting around. The cell usually goes straight into meiosis 2. So one thing that I want to mention at this point, the way that I've got this drawn, this is actually specific to a man because the 2 cells in telophase 1 are the same size. In a woman, one of them would be much larger than the other, and the smaller one would be called a polar body. It doesn't survive because it doesn't have enough cytoplasm or other cellular machinery like endoplasmic reticulum or mitochondria to be able to stay alive. After telophase 1, we go straight into meiosis 2. There isn't another replication phase the way that there was before meiosis 1 because we don't want to be recreating the DNA. We don't want to be replicating the DNA again to go through the second phase of meiosis. So in meiosis 2, we go through steps that are really similar to what we went through in meiosis 1 with a couple of differences. Prophase 1 had homologous chromosomes. By the time we get to prophase 2, we've already split those into separate cells in telophase 1, and so in prophase 2, you're not going to have you're not going to go through synapsis, and you're not going to have the synaptonemal complex because those are things that homologous chromosomes do, and we don't have homologous chromosomes. In meiosis 2, we're dealing with what are called haploid cells. Haploid cells have 1/2 of the genetic information that a normal adult body cell has, in that they only carry one version of each gene. And there's a common notation for haploid or diploid that you'll see written 1N or 2N. So here, because this is haploid, we write this 1N, or sometimes just N. In meiosis 1, most of the cells are diploid, which we write 2N, up until telophase, when the two cells divide and give off two daughter cells that are both haploid. So getting back to meiosis 2. Oh, don't forget the centrosomes here. The cell goes from prophase 2 into metaphase 2. In metaphase 2, the chromosomes line up to form another metaphase plate. This time, the microtubules attach to the sister chromatids and connect them to the centrosomes at the poles of the cell. This is so similar to metaphase 1 that it takes a lot longer to draw than it does to say it. And from metaphase 2, we move into anaphase 2, which is just going to look a whole lot like anaphase 1. Let me just be consistent with my colors here. And in anaphase 2, now instead of homologous chromosomes separating, they did the way they did in anaphase 1, the sister chromatids separate. And of course this time there's no independent assortment because these are just sister chromatids. So moving from anaphase 2, the cell goes into telophase 2, where the chromosomes will reach the opposite pole, the nuclear envelope is going to reform, the cell is going to divide its cytoplasm, the chromatin will decondense in the nucleus, and the centrosomes will disappear. Okay. So, all these cells in meiosis 2 are haploid. Even though in anaphase 2 and telophase 2 you're pulling the sister chromatids apart, you're not really changing the amount of genetic information, even though the daughter cells end up with a different amount of genetic material. Also, notice that I've drawn this for a man because the two daughter cells are roughly the same size. In a woman, one of these would be a much smaller polar body, while the other cell, the egg, ends up with most of the cytoplasm and most of the organelles. Another really interesting thing about meiosis in women is that when a female fetus is developing inside the mother's womb, the cells that will become eggs start prophase 1 and then they stop in prophase 1 until that individual reaches puberty, and then a few of these cells per month will go through the rest of the meiosis 1 cycle. That means that a woman who is pregnant with her daughter is carrying the cells that can become her grandchildren. This is called arrest. The egg actually rests again at metaphase 2 until it comes into contact with the sperm in the right way. Okay, so to recap. Meiosis gives rise to eggs and sperm, but before meiosis can start, you have to go through replication, so you have all of the DNA ready to go for the cell divisions that you need to go through. Meiosis goes through 2 rounds, meiosis 1 and meiosis 2. Each one of those is broken up into 4 steps. Those are prophase, metaphase, anaphase, and telophase 1 and 2. In meiosis 1, you have mostly diploid cells, and in meiosis 2, you have only haploid cells.